Hello, everyone. I am Eve Rannamae from the University of Tartu, Estonia. I'm a zooarchaeologist. And in recent years, my studies have focused on sheep, both from the zooarchaeological, but also from ancient genetics point of view. And I have included both ancient and modern sheep samples in my studies. So although coming from archaeology and zooarchaeology, my research has brought me to contemporary native breeds as well. And coming from Estonia, my geographical background and interests lie mostly in Northern and Northeastern Europe. Last year, I finished my postdoctoral project at the University of York. In this project, I collaborated with Urmas Saarma, Anneli Järmpalu-Idvand, Matthew Teasdale, and Camilla Spelle. So today, I would like to show you how in this recent project we applied zooarchaeology through the analysis of ancient and modern DNA to the study of contemporary native sheep breed we have in Estonia today, the Kihnu native sheep. This talk is based on soon to be published article. Few words about the study background. Here in Northern and Northeastern Europe, there is a very interesting group of sheep, the Northern short-tailed sheep, that represents an ancient type of sheep that continues to exhibit primitive characteristics. These primitive characteristics are, for example, short tail, small size, and range of colors. Historically, those breeds have been suggested to originate from populations with very long history. For example, the Soai sheep in the island of St. Kilda, Scotland, is believed to be the true relict from as early as the Bronze Age and linked to the Mediterranean and Asiatic mouflon. And the Icelandic sheep is also believed to have survived rather intact for the last 1100 years, from the time it was introduced by the Viking settlers. Northern short-tailed sheep populations have been developed and preserved in the peripheries of Europe, and are now considered to constitute an invaluable pool of genetic resources. And therefore, safeguarding their genetic diversity through conservation programs is of high importance. And of course, not only their genetic diversity is important to preserve, but also the cultural heritage and traditional sheep husbandry they come with. Supposedly, one of these northern primitive types of sheep are the Kihnu native sheep in Estonia. Kihnu breed represents the local indigenous sheep who were discovered very recently, only in the 1990s. Until then, it was believed that the local indigenous sheep here have gone extinct, that is, mixed with modern breeds in the course of time. But no, they had been preserved in the peripheries of Estonia, on small islands, and are also in small mainland regions. And so the conservation program began based on the individuals collected from two small islands, Kihnu and Mania. And in 2016, the native breed, named as the Kihnu native sheep, was acknowledged as a breed, and in 2019 as an endangered breed. It's also important to note that these two islands, Kihnu and Mania, form the Kihnu cultural space, which has also been included in UNESCO's lists, and therefore the sheep is only one part of this very important cultural phenomenon. Now that I've briefly introduced you the importance of Kihnu native sheep and the role in a very unique cultural space, then why do we need to study this breed further? Despite the recent acknowledgement, the position of the Kihnu native sheep among the rest of the northern sheep population has still been under discussion. Although the primitive characteristics are exhibited in Kihnu sheep as well, the tail length, for example, has been debated. Until now, some previous genetic analysis of microsatellite markers and mitochondrial DNA have indicated that the Kihnu sheep are genetically distinct. They are divergent from modern breeds and affiliated to ancient populations. But more genetic analysis have been needed to clarify the origin and breeding history of the Kihnu native sheep. And so in this study, we contributed to this exploration through the examination of endogenous retroviruses. Since I'll have a short talk today, I won't tell you about endogenous retroviruses in detail, but as you probably know already, they have shown to be a good marker for studying the origins and population history of sheep. Among these studies, the most central example for our study was the one published by Chessa et al. 11 years ago. There, they show two major migratory episodes of domestic sheep. The first migration out of the Near East, the genetic signature of which has been preserved the primitive groups in the peripheries, the feral mouflon and northern short-tailed sheep, and later a second migration of improved wool sheep, possibly also out of Near East, which laid the foundation for today's modern breeds. So very simply, we wanted to see what would the retrovirus markers show for the Kihnu native sheep. Would Kihnu sheep have the same retrotypes that are in other Northern European sheep? And to better see the development of those markers in Estonian sheep populations in general, we included ancient samples as well. Methodically, we followed the same protocol as was published by Chess et al. In 2019, with slight modifications. We focused on 4 independently inherited insertionally polymorphic endogenous retroviruses, which have shown to be the most informative retroviruses. Using the technique of polymerase chain reaction to search for and amplify the necessary DNA markers, we confirmed whether the retrovirus integration in the genome of each individual was present or absent. And based on the combination of this presence-absence, we assigned individuals into so-called retrotypes. And then we compared the frequency of each retrovirus and retrotype between the Estonian ancient populations, Kihnu sheep, and the selected reference data. We received positive results for all 80 Kihnu samples and 53 ancient sheep samples, which is a very good success rate. To evaluate genetic relatedness, we compared frequencies of each provirus in the analyzed groupings, and our study revealed that yes, there have been changes in the retrovirus diversity since the late Bronze Age to present day. With this table here, I want to show you these frequencies and changes in our study populations. I won't go too much into detail, but just so you could follow me, here we have the ancient populations separately by each time period. We have ancient total here and Kihnu total here. And following these, we have the reference populations of northern short-tailed sheep by country, Russia, Finland, Sweden, Norway, Iceland, Faroe Islands, and Great Britain. And in the very end, we have 4 improvement breeds that have also been used to improve the Estonian modern sheep breeds, the Estonian blackhead and whitehead sheep, starting from the 20th century. Here in gray, you can see the tested number of individuals for each provirus. And in these columns here, we have the frequencies of each provirus. And based on these frequencies, I will quickly now explain our study results. I have highlighted in yellow the rows by period, so we could have a better overview of the numbers. The frequency of B7, which is the oldest provirus and is indicative of primitive breeds, as you can see, has been decreasing since the late Bronze Age, but it is still present with a rather good proportion among Kihnu individuals. The frequency of B18, however, which indicates a more recent ancestry, has been increasing in time. It seems that this increase is especially visible starting from the Middle Ages and early modern period. And according to Chess et al., the higher frequency of this provirus characterizes the wool sheep from the second migration wave. Therefore, maybe the higher frequency in Estonian populations could reflect the introduction of improved lineages, which coincides with the expansion of the medieval trading network and the beginning of early modern breed improvement. The frequency of 5 of 16 is the least informative, since it has been quite constant from the Iron Age to modern period. But the 4th provirus, B8, is quite interesting because it appears in the tested sheep populations only in modern times. This provirus was not present in Estonian ancient sheep and also in many of the modern northern reference breeds, but it is present in Kihnu sheep. In Kihnu sheep, almost 1/3 of the tested animals had it, which is similar to Texel and Leicester breeds. Therefore, the presence of this provirus in Kihnu populations could result from the 19th, 20th century improvement of the local animals with these particular imported breeds, or perhaps from the effects of recent inbreeding or genetic drift. Now, if we come to the retrotypes that are combinations of these 4 tested proviruses, then out of the 15 described retrotypes, 13 were found in Estonian sheep. Temporal view on the assigned retrotypes showed that 8 retrotypes of these were present in both ancient and modern populations in Estonia, while the remaining 5 were present only in Kihnu native sheep. Let's quickly go through these retrotypes. Retrotype 0, which is in white, and 1 in red, have been described as the most primitive ones that are prevalent in northern short-tailed. And we were very happy to see that they were also present in the Kihnu native sheep, and of course also in ancient samples. Neither of the 2 retrotypes occur in the analyzed improvement breeds. The most common retrotype, retrotype 2 in green, is associated with the secondary introduction of improved wool sheep from Southwest Asia. It appears to be absent in the most primitive breeds like Soai and Orkney, and it was also absent in the late Bronze Age individuals in Estonia. R3 in darker blue has been referred to as Nordic retrotype and is widely present in northern short-tailed sheep. Our results show that also Estonian sheep have retained this retrotype since the late Bronze Age. Although its frequency has decreased significantly in Kihnu native sheep, we still detected it in 2 individuals, while in the ancient population it was more prevalent. And other ancient retrotypes that were present in ancient sheep samples, retrotype 4, 5, and 6, were also present in Kihnu sheep. Now, retrotype 7 was interesting because although we found it only in 1 ancient individual, the radiocarbon dating placed this sample to early modern or modern period, probably 17th, 18th century, but more probably even to the 19th, 20th century, which was the time of large-scale breed improvement. Therefore, it seems that R7 is likely a modern introduction related to recent breed improvement. What do our results mean? First of all, and the main result is that Estonian ancient and modern Kihnu native sheep share the same ancestry with the northern short-tailed. If you come back to the background information once more, then previous studies have suggested that in Northern Europe, there was an initial founder population that fragmented into isolated populations, who today we know as the northern short-tailed sheep breeds. Over the course of time, these peripheral northern populations became distinct from modern breeds, but at the same time also lost their inbreed diversity. This loss of diversity seems to be reflected in our results of the provirus integrations as well, because our analysis of the retroviruses and their combined retrotypes in ancient populations, especially in the late Bronze Age and Iron Age, indicated often a wider diversity of retrotypes in the past than we see in the native northern short-tailed breeds today. And this means that the frequency of primitive lineages in the northern short-tailed sheep seems to have been decreased. Second result was that overall we saw different fluctuations in the presence and frequency of different proviruses and retrotypes. These fluctuations seem to reflect the overall development of local sheep populations influenced by historical factors like trade and the 18th, 19th century shift to modern breed improvement. And the third important result was that when looking at Kihnu native sheep, then they had a very high diversity of retrotypes. This could reflect the twofold ancestry of the Kihnu native sheep, the presence of the oldest proviruses and primitive retrotypes that originate from ancient ancestry, together with novel retrotypes resulting from introgression with improved breeds. Based on the retroviral integrations analyzed in this study, it seems that this kind of crossing has also sporadically occurred in some other northern short-tailed breeds. Unfortunately, it is impossible to say whether this introgression first happened in the distant past or more recently. Importantly, however, crossings with modern breeds seem not to have been too extensive because several lineages carrying the oldest proviruses and ancient retrotypes have been preserved in the Kihnu native sheep breed. In light of our molecular results, Kihnu native sheep truly seem to be the descendants of the past populations in Estonia. And all those ancient retrotypes that have their ancestry in the past populations are worth preserving. Selection strategy by the Kihnu Native Sheep Society focuses on previously established genetic lineages and on the presence of indigenous traits, especially regarding horns and face. But now they also have one more aid to the selection strategy. The individuals carrying the 7 ancient retrotypes, retrotype 0 to retrotype 6, are now used and selected for in the breeding program, especially those carrying R0, R1, and R3, with an aim to preserve these northern primitive lineages. Attention is also given to morphological traits that comply with the retrotypes. Interestingly, desired indigenous phenotypic traits seem to be in accordance with the presence of primitive retrotypes, but of course, this is just a visual observation for now. So all in all, sheep with morphological features characteristic of the Kihnu breed and now also with suitable retrotypes get more points in the breeding program and are more valued breeding animals. Here on this slide, you can see a few examples of the Kihnu sheep who represent the 7 retrotypes valued in the breeding program now. You can see the very indigenous characteristics like small size, horns in both males and females, clean face, glossy faces in white sheep, short tails, 6 different color types, changes in colors related to age, presence of wattles, double layered coat, and slim legs. One of the features the Sheep Society is interested in and regarded as a very specific characteristics to Kihnu native sheep are the Dalmatians, the Dalmatian color type. Dalmatians among the Kihnu native sheep seem to have a specific feature of very clear patches and with white patches that turn black spotted with age. To the Society's knowledge, these features are not characteristic to other Scandinavian sheep. And this is all from me. Thank you for listening.